afternoon, everybody. My name is Charlotte, and I should start by, as most people do, thanking the organisers for asking me to speak today. I have realised that my abstract was a bit ambitious to squash into 20 minutes, so there's going to be quite a lot of squashing going on here, so I'll do my best to try and talk to time. I'm going to talk about a few ways of trying to improve fragment assembly protein structure prediction. Now, I'm sure most people in the audience are very familiar with fragment assembly protein structure prediction. It's been used as a sort of common technique for quite a long time now. The basic idea is I have a sequence, and I'm going to build it from lots of pieces of known structure. So it's a bit like building a Frankenstein's monster, because what I'm going to do is rip pieces of protein structure and stick them together and hope that what I get looks like another protein or looks like another human. The big differences between building Frankenstein's monster and building a protein is I don't know what the answer should look like. So it is completely possible for the position where I think there should be an arm that I have no arm fragments at all. I just have lots of other different fragments like multiple legs. So I could produce something that never looked like the correct answer. So what I have are lots of possible fragments and under some form of energy function that might include, for example, the contacts that were mentioned earlier in the session. I then try and fold it up into a three-dimensional structure. So the first way I'm going to talk about improving this is thinking about those fragments that go in. If you don't have the right fragments, you can't make the right protein structure. Okay, there's no possibility, you can't actually get there. So somehow within the set of fragments you have to have the right shapes. So fragment libraries are quite simple in concept. What you have is a section of the protein, so it's a segment of residues that you're interested in predicting, so say this red boxed area here, and you collect a set of fragments that you think at least one of these will be the right shape for that. And the way you collect those is scoring usually on the similarity of sequence between your segment of residues and segments of known structure. And you just repeat this. So then you have blue set of bricks for the next bit, green set of bricks for the next bit. So you just keep going on and on. Now what you'll notice, even in my pictures of bricks there, that for some places what you'll end up with is lots of fragments that all look very different from one another. So you can imagine if you were looking at, say, a loop region of a protein structure, that's something you'd expect to happen. It's very difficult to know what that shape would be, so when you're trying to work out what it might be, you'll put in lots of shapes that are all very different from one another. Or you can imagine if you're in that blue section, maybe that's a bit like an alpha helical piece. So if a part of a protein structure is predicted to be alpha helix, then pretty much every fragment you're going to put in is going to be an alpha helix, and pretty much every alpha helix looks like every other alpha helix. So you'll have a set of bricks that all look very, very similar to one another. So we set about writing a fragment library method to generate those kind of pieces that took that into account. So this is just one piece of data on that to give you an idea of what it looks like. So there's a scoring system running on the x-axis there. So this is actually just a Ramachandran-specific sequence score, so it's based on the predicted Ramachandran angles for these. And then you're saying the proportion of the fragments that I can get out that would actually be any good is my bar chart. And the proportion that I've actually managed to cover with a good fragment is the red line there running across the top. What happens if I'm looking at alpha helical fragments, I can get a good fragment almost every single time, and almost all of my fragments are good. But if I'm looking at a fragment which I think is coil, so loop down in the bottom right-hand side, there's almost no correlation between my score and whether a fragment's going to be any good, and it's very hard to have any good fragments there at all. So it turned out that what you needed to do was use separate scoring systems for the different types of predicted secondary structure. Now, I was going to keep talking about how we did this for the next sections of the slides, but I'm now going to slightly change track, because since we published our paper, lots of other methods for doing fragment library generation have come out. And this is great, there's lots more diversity in the field. So our method is the one called FLIB, which is the orange line on these. And we had compared to several other methods, but primarily the sort of bright green line, which is NM-make, which is the method used by Rosetta. And you can see, if we look at precision, we were a bit better than NM-make, and coverage, we were about the same. Now, so a couple of methods came out, FragScion and ProFragger, which were designed for being very rapid. Now, their results are not as good, potentially, but they're very, very fast, so they have other utilities. And then a method came out called LR-FragLib. And we looked at it and we were like, okay, so this is the olive green line on here. This is much more precise than any other method out there. And we should use their method. they've clearly got this right. So we did. And this is what happens if you make models using those different methods. So I'm making three-dimensional models now, and these are just the best models that come out of the system. This is just a set of targets running across the top, and then these are the different fragment libraries used as input. Everything else is identical, so the PDBs they could have used, everything like that. And what you'll see in orange is where that method produced the best, and in blue is where a method produced something that was almost as good. And you'll see that the best models are always produced either by our own fragment library or by NMake, and not by lrfraglib. And we were a bit like, but they've got much better fragments in, so theirs should have produced the better models. And this seemed to be something that was worth investigating. So we knew it had better fragments in, we checked the precision, but it wasn't producing a better model set. So we went back and looked at the predominant secondary structure type. And this matters, and it turned out it mattered a great deal. So if you look at the graphs for the secondary structure type now, this is the same graph of precision, but just split by the predicted secondary structure for the different fragments. So all of the methods do very well for alpha helices, and the best is lrfraglib in the sort of olive green. But then in the other types, it's nowhere near as good as some of the other methods. So why was it overall more precise? Well, because it was doing something that's intelligent if you had trained yourself against the global precision graph. It predicts lots of Lego bricks, lots of pieces in alpha helical regions, and it predicts very few pieces in loop regions. So if you check if the fragment library is precise, the answer is yes, because the percentage of fragments that are close to the correct structure is very high. But actually, it's kind of doing the reverse of what maybe you would want a fragment library to do, because probably, and actually none of these methods yet really do this, you should have more fragments in the loop regions where it's hard to get the Lego brick right, and you should have fewer fragments in an alpha helical region where pretty much every brick will be right. So I am making a small recommendation when you are considering fragment libraries that you should look at their behavior under different secondary structure types, and you should look at how this might affect the global precision that is being given. So into the second way of improving fragment assembly, and this is really to think about improving your search strategy. Now we've had some talks about folding already in this session, and a lot of that has been about the folding that we can observe, if you like, in the test tube. The part of folding I'm going to think about is potentially the folding that might be happening in the cell, and whether that can help us with our ability to predict protein structures. So there is a hypothesis that proteins begin to fold as they're being synthesized. This is known as cotranslational protein folding. Now, there are many reasons why this hypothesis exists. The big ones are basically that we know that proteins can fold much faster than they are translated. So unless you think that the protein is being translated and then sitting there and waiting, it's definitely doing something as it's coming off the ribosome. There is also a lot of experimental evidence. So there are cryo-EM structures of the proteins partially folded attached to the ribosome. There are active proteins when they're only partially translated. So it is likely that many proteins are folding as they are translated. So what would that mean if I wanted to put it into protein structure prediction? Come on, there we go. So normal protein structure prediction would look like this sort of video on the right. What I've done is I have the whole chain there, and I'm trying to put in these different fragments all the time. Cotranslational protein structure prediction would look like the picture on the left. So every so often, you'll notice the chain gets a bit longer, and then fragments trying to put in anywhere. So it's not that only the fragment at the end would move, everything can still move, but I'm basically solving a series of local optimization problems, one after another, rather than a big global optimization problem. What that means in terms of doing protein structure prediction is that, of course, if you're only having to solve local, smaller optimization problems, each one is faster. So the scoring functions that are used for protein structure prediction are mostly sort of n squared with the number of atoms that you have in your protein. So if I have less atoms most of the time, because I'm working cotranslationally, the optimization can be done faster. But there is obviously a problem there, that if this local optimization doesn't work, I could get caught in traps which are inaccurate. So they would mean that I would never get to the correct fold because I would be stopped along the way. So the first thing we did with what we hoped was this faster method was try and work out how many decoys we actually needed to make. So how many times do we have to run this? How many structures do we have to generate? And we had a look at lots of papers for the numbers of decoys that people were producing with these methods. And one of the things I found quite interesting is that actually most people haven't done this analysis. So they just say, make as many as you can, get your biggest computer and run it, and run and run and run, and if you can run some more, that's great, and then look at what you get out the other end. And so the numbers varied a great deal between different methods. But there wasn't a sort of consensus that if you generate more than 1,000 decoys, it will be good, or more than 100,000. There was no idea of when it would be good to stop. So we took just over 40 different test cases and produced 100,000 decoys for each one. And then we took samples out of that 100,000, so say samples of 5,000 decoys, and looked at the proportion with at least one correct model. So I'm using the same definition of correct model as was mentioned earlier in the first talk in the session of a TM score greater than 0.5, but it doesn't matter a great deal, it's just a definition. And so then we could say, if I do that, how many models do I need, how many decoys do I need to generate to say that 95% of the time I'll have at least one correct model in my sample. You could do the same thing, and I can show the results for it, for the best model. So how many do I have to generate that I will be within a very small distance of the very best model I have in the 100,000? And this is what the results look like if you look at the number of decoys required to produce a correct answer. Now, you'll notice there are less than 40 proteins on the x-axis here, because I'm only looking at those cases that within 100,000 actually gave me a correct answer at all. So like all of these methods, nothing is perfect here. But in our hands, it took about 10,000. So not every time. You'll see there's 5 points that are above that blue line for 10,000. And it turned out it was about 20,000 decoys to get something that was very close to the best answer you would get in 100,000. The other thing that we found was this wasn't dependent on length. So if the protein was less than 250 residues long, it was effectively, this is what it looked like. What's interesting about that for us was that we kind of thought it probably would be very strongly dependent on length. It may well be beyond the 250 residue barrier. The problem was that we barely have any proteins we got right that were longer than 250 residues, so it's very difficult to be able to say anything about that. So this is all fine, but maybe it's all pointless if, of course, global folding is better for you. In other words, if you're allowed to make moves everywhere. So if my local moves are going to trap me in conformations that are impossible, then this is obviously a very bad strategy. So this is to give you an idea of whether that happens as well. So on the x-axis is using our predictor and using it in a directional sense. So that is like the red folding I showed you earlier. So I'm slowly extruding the protein, so I'm folding the first 10 residues, then the first 12, 15, and growing it and adding fragments and adding fragments. And on the y-axis is doing it globally. Everything else is identical, so it's the same fragment libraries, the same function, energy function they're being minimized under, the same number of moves are used, so there's no kind of bias towards either. The scores on the axis are TM scores, and any point below the line would mean that going sequentially, so going in this co-translational direction, is doing better than going in the non-sequential direction, so just going non-sequentially, so doing the whole thing all at once. And you can see that nearly all of the time, it is better to go in a co-translational direction. So not only is this method faster, but it is also doing better in terms of actually giving you a structure prediction, so you're more likely to get a correct answer. The two different graphs here, the one on the left is for soluble proteins and the one on the right is for membrane proteins. And I should say our method is really not optimized for membrane proteins, so those results are not great. So improving the search in this way, in this co-translational protein structure prediction way, is really thinking about the fact that currently de novo structure prediction methods randomly sample protein conformations. And that does require large amounts of computational resource. It's a computationally intensive problem. And earlier in the talk, they were talking about the fact that, you can't do large-scale analysis sometimes using some of these methods because it's too slow. So thinking about ways that can make it faster are clearly very advantageous. The other part of that is that, of course, you can make it, if you can make it faster, you can generate more decoys. So the other way I could have done this was have used the same amount of computational time for those two axes, in which case the co-translational ones would have been able to generate more decoys and would probably have performed even better because they could explore more of the space. The strategy we're using is suggested by biology. Now, that doesn't mean that this confirms the theory of co-translational protein folding, but it is a relationship between how biology is doing this in such a fast and efficient fashion. So I suspect that there are other things that are suggested about the ways proteins fold in a cell. So, for example, they fold into a crowded environment. They fold against a very large object called the ribosome. And these kinds of things may also be ways that you can reduce the space that has to be sampled and therefore speed up the way you do it. We require about 10,000 decoys to be certain that we're going to get a good answer. And that is fewer than most methods are suggesting are required. So I think this is a good thing. In our hands, this improves speed about 1.5 to 2.5 times. I think probably, the actual speed up depends exactly on how you would perform the extrusion. So it should be about 3 times faster if you could do it perfectly, but obviously we haven't quite done that. Reassuringly, or perhaps better than that, the sequential procedure produces better models. So you're producing a better answer by doing this. So I am not getting trapped in local minima by making this choice. So I think it's a pseudo-greedy search strategy. That's all I'm doing, and it reduces the computational time to generate de novo protein structures, and it actually improves their accuracy. So I should finish by acknowledging my group, but most particularly this man here. So this is Saulo, who actually did all of this work, and should probably have been standing here giving this talk, but he's attending a friend's wedding today, so I am here instead. So thank you very much.
Nice talk. Did you ever try it from the simulation from the C-terminus folding to the N-terminus just to see, is it an evolved property of the sequence?
So yes, we did, and that has different behavior depending on soluble and membrane proteins. So in soluble proteins, we still see the improvement is more N to C, but C to N is still better than global. With membrane proteins, we actually don't see any difference depending on which direction we run it in. So C to N is as good as N to C. So I don't have a complete answer to that, but we have done that as well.
Hi, Charlotte. Great talk. So there seems to be this really 2 alternate universes in which people in the structural biology community are living. One is the sparse evolutionary fragments from the previous talk, and one is the utilitarian, let's do better protein folding fragments kind of in your talk. And my question is, do you think you guys can ever meet and do things together in that respect and probably solve life, the universe, and everything and while doing it?
I think there's a general kind of movement where that's starting to happen. I think what evolved were 2 very separate communities that thought about protein folding in one space and protein structure prediction in the other. And I think that there is a movement at the moment where those are starting to become more related again. I think they were originally very strongly related, but it became the ways that were successful at protein structure prediction did not relate so much to what was observed.
It's more like protein evolution and classification on the one hand and prediction on the other, I think.
So I think evolution and classification feeds into all of this quite easily because that's related to how you might select your fragments, what kinds of fragment libraries you would build, the kinds of information that you would include in your scoring systems. So I think lots of that has fed into these kinds of algorithms, in lots of other people's work. So I think that's happening.
Thank you very much.
So I have is this working? Okay. I have a technical question about the method that you presented. In a sequential greedy algorithm, you have to make certain decisions about, say, the length of the window or what it is that you call it for a sort of a self-folding unit where you first generate something and then I guess you decide to grow it, to stop exploring for this window and then grow it. How do you make this decision? Does this affect the quality? When do you know, so how big should the window be? And then when do you know to stop exploring for this window and growing? Because one of my concerns is you have long-range interactions. Imagine a beta hairpin that has to come back. If your window cut just in the middle of that, you're going to get some fault. How do you undo it, right? You just got it wrong. How do you keep improving?
So that was my point about a genuine concern is that this kind of method could get you trapped in local conformations, so local minima, but that doesn't appear to be happening, otherwise we would not be performing so well. In terms of the choices, at the moment we have it in a very straightforward way. We have played with these a bit and not seen massive differences. So the extrusion is actually based on the way the fragment libraries work, and our fragment library, like most, has a kind of standard length which is about 9 residues long. So you have to start with 9 residues. We actually grow a single residue at a time, so when we're growing, and we have a kind of linear number of moves that occur throughout. And we just took the number of moves that we were using for our global folder and divided it across, minus the last 1,000 moves, which we just use on everything at the end as like a tidying up phase. So there's nothing sophisticated about that process. I mean, you could have something very sophisticated there because there is some knowledge of the speeding up and slowing down of how proteins are produced by the ribosome. So if you wanted to be very sophisticated, you could say, we know there's a pause now, so we would give longer for this to try and fold before the next bits come. But we haven't done anything that complex yet.
Okay, last question.
Charlotte, you mentioned just now that, is it working?
It is now.
Okay. Okay. You just mentioned that the size of your fragments are on average 9 residue long.
Yeah.
Does that change according to the type of structure that they represent, or how is this distribution?
So we do have variable fragment length in our fragment library, but the distribution of lengths is pretty much identical for the different secondary structure types. So there isn't sort of alpha helical fragments being much longer and loop fragments being much shorter. I suspect that there are far more intelligent things that could be done there than what we have done. So I think that probably, there should be more variation in those kinds of things, but we haven't done it.
Okay, let's thank Charlotte for the nice presentation.